Okay, if we go back to some Mendelian genetics terms here, this is actually the beginning of part 2. Sorry about getting so close into it, or quickly into it. Mendelian genetics 2. I'm going to kind of fly through this page here. Remember we talked about characters and traits. Characters are general characteristics that are shown in organisms. Traits are different forms of those characters. Traits are controlled by discrete units on the DNA called alleles. And these alleles can be dominant or recessive depending on the specific allele. Remember, the alleles are specific places on chromosomes. And these here are homologous chromosomes, meaning they have the same type of information, but they don't have the same exact information. In this example, we have two different alleles. All right, we can have the two of the same ones if we want. We can have two of these, or we can have two of these. Whether or not these are dominant or recessive is manifested or shown when these two alleles are present together. Whichever one shows the phenotype is the dominant allele. The genotype, remember, is the combination of alleles that you have. All right. So if we were to look at this plant right here, this plant has a big P allele, which is purple. If we go back, I didn't say it there. This organism has a genotype combination of big P, small p, but their phenotype is purple flower color. Whereas this plant here also has purple flower color, but it has a different genotype. So phenotypes are the physical characteristics or visible traits, and the genotypes are the allele combinations. All right. This here is a Punnett square. We're going to talk about them later on. It's just showing you the possible characters. Remember, each parent has two forms of each allele and can only pass one of them. All right. This organism is homozygous because they're the same two alleles. This organism here is heterozygous for purple. This organism is homozygous for white flower color. As you can see, they have two recessive alleles. This here is called a monohybrid cross because we're only crossing. Actually, this is not a monohybrid cross. I apologize. This is a monohybrid cross right here. We're taking a true breeding purple plant and we're crossing it with a true breeding white plant. The true breeding part and the pure breeding part means that this organism only has alleles for purple and this organism only has alleles for white. And we can create those by just mating white plants with white plants and taking out any purple plant alleles and getting rid of them, and continuing to mate the white, 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 and white. And at some point, we're going to eliminate the possibility of the purple allele in the population. All right. A monohybrid cross is where we're taking one character, in this example, it's flower color, and we're crossing two true breeding organisms to see what their offspring look like. Now, a dihybrid cross is basically, let's see here. No, sorry, I'm not showing a dihybrid cross right now. But if you can imagine, this is just flower color. If we were to cross an organism that is true breeding for purple and true breeding for height, like big T, two dominant alleles, and crossing it with a white short plant, then that would be an example of a dihybrid cross. The P generation is just the parent generation. We're crossing two organisms. There would be the parents. Whatever their offspring are is called the F1, or the first filial, first sons, first daughters. All right, if you take Latin, you might know that. So the first generation is the F1. If we were to take offspring from the F1 generation and mate them to create another generation, we'd end up with something called an F2 generation. All right, which is the second generation. Okay. So just some terms that are important to know. We're going to get into all this stuff more discreetly. But one thing I'd like to talk to you quickly about, if an organism, remember, homologous chromosomes have the same type of information, but not exactly but may not have the same information. Right? So if I receive this chromosome from my mother and this chromosome from my father, what I see are two different alleles. These are for a character. That is called heterozygous. If I have the same two alleles for a specific character, I'm called homozygous. Sometimes an organism only has one allele, and this would be an example in males if specific genes that occur on the X chromosome are not on the Y chromosome, then those males will only have one allele because the allele may not be present on the Y chromosome, and we'll talk about why that is later on. This organism would be called hemizygous. Okay. Okay. Now, Mendel's first cross was called a monohybrid cross. All right. So the first thing he did was he took true breeding tall plants and he mixed the pollen of the flowers from the tall plants and he sprinkled them on the female parts of the short plants, and he looked at what the offspring were. And he found that in the next generation, he found all tall plants. All right. So he took two true breeding plants for tall and short, and he mixed their alleles together using cross fertilization, and he found that the F1 generation was all tall. Now this showed him something. This showed him that the trait for tall was dominant over the trait for short. Because this plant can only give alleles for shortness, because it's true breeding. And this plant can only give alleles for tallness, because it's true breeding. So if you mix one tall allele with one short allele, you're going to get two alleles. He didn't know exactly this yet, but he did know that the trait for tallness was dominant over shortness just by this first part of the experiment. All right. Now, he only had tall offspring. All the offspring were tall no matter what. So no matter what, all the offspring were always tall. Then what he did was he mixed two tall plants. He took the flowers from our F1 generation, all right, which were a tall plant, and he crossed it with another tall plant. Not a true breeding tall plant, but another F1 tall plant. All right. And what he found was that the offspring were some were tall and some were short. Now, the question is, what do you think this led him to think? All right. Well, this supported his idea that one trait must have masked the presence of the other, but there was something in the short plant that was here that wasn't showing itself but could be passed on. All right. Now, remember that this organism is probably a heterozygote, meaning it probably has an allele from the shortness, it probably has an allele from the tallness, but the tallness allele is masking the short allele, so this organism might be heterozygous, but even though it's showing tall, maybe tall is the dominant phenotype. So when this organism was mated with another organism similar in the F1, it resulted in some organisms having the short phenotype. All right? So this was called his monohybrid cross, and he specifically found that organisms in the F2 generation of a monohybrid cross, this whole thing is a monohybrid cross, all right, the F2 generation was showing a ratio of 3 tall to 1 white, or dwarf plant, or 3 tall to 1 short plant, and that was the ratio. Even though it might have been like 3,211 to 999, he still found a 3-to-1 ratio always. All right. So, if we back up here, this is an example of a monohybrid cross too. All right. So what we're showing here is we have a true breeding purple plant with a true breeding white plant. All right. The two alleles of a character, which are flower color. When this organism can only donate one of these peas, and this organism can only donate one of these peas, all right. So we end up with offspring that are all big pea, small pea. All right. But if we take these organisms and we cross them, all right, we have a bunch of different allele combinations that can be shown. So we're talking about the monohybrid cross here. Let's go to the next one. Let's talk about these tall plants, and here we go. Okay, so let's assume that in this monohybrid cross, we're doing one character, which is what Mendel did, and what's in that character is height. All right. So in that height, we have tall plants and short plants, and we just established here that one of them is a dominant characteristic and one is recessive because of our F1. So let's say that Mendel already knew that, even though he didn't. All right. So our tall plant, if our tall plant is the dominant phenotype, then that means that our tall allele is going to be this one, and our short allele is going to be this. Now, how did I know that? All right. Well, we just established that the tall allele we said was the dominant phenotype. So its allele is uppercase T. And if the short is masked in the presence of the dominant, then it's the recessive allele. So it needs to be denoted with a small t. All right. And remember, each parent is only going to donate one or the other. Now, so we have big T, small t. All right, we've established that. Now, we go back to the parental cross. He took a tall plant, a true breeding tall plant, and he crossed it with a true breeding short plant. All right. Now, each parent is only going to donate one of these. All right. Remember, during meiosis, each sperm cell is only getting one form of each character. All right. So each sperm cell holds one of every single possible gene that there is for that organism. All right. And then the female, she in the egg cell, it has one of every possible gene. Now we're just talking about one character here, not the hundreds and thousands that we have. We're talking about one. All right. Is a monohybrid cross. So we have true breeding tall and true breeding short. You could imagine that this organism can only donate a big T because it's all there is to give. It has to donate one of them. And this organism can only donate a small t. That doesn't look good. All right. So all the offspring are going to be heterozygous. Now, if we can do something called a Punnett square is basically going to show us parent 1 goes on one side. We can donate this or this or this. And this parent goes on the other side. This parent can donate either a small t or a small t. All right. Now what we do is we match them up. Okay. So let's do that for a moment. This organism can donate big T, and this one can donate a small t. We can donate a big T. We can donate a small t. We can donate a big T. We can donate a small t. We can donate a big T. We can donate a small t. So all of our offspring here are going to be heterozygous, showing the tall phenotype. Does that make sense? All right, that's all the F1 generation. All right. Now, so we know that no matter what, where are you? Throwing that away. No matter what, all the offspring are going to be big T, small t. Bam! All the offspring, all the F1. So what Mendel then did was he took all the F1s and he crossed them with each other. So he did in the F1 generation, he took a tall plant and he mixed it with a tall plant. Now, he did not know that these were the genotypes, all right? He didn't know that, but this is what they were. This is what they had to be, because the short plant was only donating this allele, the tall plant was only donating this allele. So all the offspring were the same. He didn't know the genotypes yet, all right? Then he crossed these two in the F1. And again, we do our Punnett square. We can donate this or this. And this organism can donate this one or this one. So if we match these, this is a possibility, this is a possibility, this is a possibility, and this is a possibility. All right, we end up with his 3-to-1 ratio. Okay, we end up with his 3-to-1 ratio. You can see the bottom now. I forgot to show it in the last video and I'm redoing it. But what this shows is that it's possible 1, 2, 3 are showing the tall phenotype and one of them is showing the recessive phenotype. All right. So the chances of getting a tall
offspring from the F1 cross are 75%, and the chance of getting a short organism are 25%. All right, we're going to talk a lot more about this in class, but hopefully this clears things up just a little bit for you. All right. The second thing he did was he did something called a dihybrid cross. All right, and we're going to talk a lot about dihybrid cross in class. What I'd like you to do is actually look up Mendel's dihybrid cross. And it's basically the same thing as a monohybrid cross, except he's using 2 characters instead of 1, because he wanted to see if these characters are sorting independently or not. So he took like a pure breeding tall purple flowered plant and he crossed it with a short white flowered plant with the recessive alleles. All right, and he basically did the same thing, and we're going to talk about what his ratios were. All right, but that's Mendel's monohybrid cross, and that's Mendel's dihybrid cross. All right, so yeah.